Okay, so
Hard to get it.
On 76 interface. That's
oh yeah, okay, so we're talking about cells, right? Parts of the cell and functions of the cell organelles. We talked about the plasma membrane and how atoms and molecules get into and out of cells, which happens all the time. Cells are always moving stuff in and out. Some stuff comes in and out by itself: oxygen, carbon dioxide, water. Okay, and we talked about the general structure of a cell and some of the major organelles. So, talk about all this stuff. The nucleus, chromosomes. Structure of DNA, which is what our chromosomes are composed of. So talked about all this stuff. Any questions about this stuff so far? So we were into this stuff, right? Cell cycle. Okay. So the cell cycle, this term cell cycle, just means the life of a cell, the stuff that happens during the life of a cell. And we consider the beginning of a cell's life is when it is formed by a parent cell dividing. So every cell had a parent cell, and then that parent cell divided, and now we call these the daughter cells. And so this is when we say that this is the beginning of the life of this cell, when its parent cell divided. Now, its life will last until it divides. And then we will consider that to be the end of that cell's life. Now there are two daughter cells. So the life cycle is from this division to this division. That's what we call the cell cycle. The cell cycle consists of can be divided into two periods of time: interphase and the cell division. And most of the cells in your body, we call them somatic cells. The only cells that are not somatic cells are cells in your reproductive organs, in the testes of males or the ovaries of females. The cells that divide to produce sperm and egg. So those cells that divide to produce sperm and egg, and also the sperm and egg, those are reproductive cells. All the other cells are somatic cells. And so there's two kinds of cell division that we're learning about. One is called mitotic or somatic cell division, and that's what all those somatic cells. It's how they divide to make more cells. When we grow, when we heal, some cells get destroyed and they have to be replaced. They'll be replaced by mitotic or somatic cell division. Some cells just have a short life normally, and so they're constantly being replaced or regenerated as the old ones die. Red blood cells, skin cells are like that. They don't last very long, so we're always making more. By somatic cell division, this reproductive or meiotic cell division only occurs in the reproductive organs, only in the testes of males and the ovaries in females, only for making sperm and egg cells. Those are called gametes, or sperm and egg cells. The process of cell division is similar in both types, but a little more complicated in the reproductive or meiotic cell division, because there's actually two divisions. The parent cell in the testes or ovaries will divide, and then these daughter cells will divide again. That's in the reproductive or meiotic cell division. In the somatic cell division, only one division. Anyway, so this is the diagram of the cell cycle. Yeah, this one's more detailed, shows more details, but the G1 phase, G stands for growth, S stands for synthesis. Synthesis means making something. In this case, it's making DNA, copying the chromosomes will cause the cell getting ready to divide. So it needs to double the number of chromosomes so that each of the daughters will have as many as the parent cell had. So that's all interphase. G1 phase, S phase, G2 phase. So growth after the cell is formed, so it becomes the normal size. And also, if the cell is getting ready to divide itself, then in the S phase it will replicate the chromosomes, and then the G2 phase, the cell will grow some, maybe make more organelles, so that both daughter cells will have enough to get started in their new lives. This G0, this arrow going off to the side, called G0. This is when the cell is not dividing. It's not even thinking about dividing. It's just living its life as a cell in your body. So it could stay in that G0 phase for years, or some cells stay in G0 phase forever, because they never divide once they're formed. But some cells might stay in the G0 phase for a long time, and then for some reason they are stimulated to divide, and then they'll go back into the other phases of interphase, and then they'll go into this one phase where they divide. The M phase, mitotic phase, can be divided up, as you see, into 4 phases: prophase, metaphase, anaphase, and telophase. Just remember PMAT, P-M-A-T. If you include interphase, then it's IPMAT, I-P-M-A-T. So we have to know a little bit about what happens in these 4 stages of mitosis. So the word mitosis in is it's really mitosis is really the just division of the nucleus of the cell. We some people use mitosis to mean cell division, but it's actually correct. The correct use of the word mitosis is that's only the division of the nucleus. So the division of the whole cell, that by this type of division, we should call it mitotic cell division. So the mitosis is really only the nucleus part. Um, so this is a series of diagrams that summarizes what happens in each of these phases. This one, this I don't know why some books do this, but they have some books have an extra phase called pro-metaphase, like in between prophase and metaphase. Most books just have prophase, metaphase, anaphase, and telophase. Some books have to be weird and put an extra stage in there. I don't know why you can't all do the same way. But it's just the same events happen, um, no matter how many phases we consider. But first of all, before the cell divides, it has to copy all of its chromosomes so that there will be enough for the two daughter cells. When a cell is not dividing and not getting ready to divide, the DNA of the chromosomes it's all just stretched out, or it's not coiled up tightly as it is in these visible chromosomes that you can see under a microscope. So during interphase, you can't see chromosomes under a microscope because it's just DNA molecules. They're not coiled up to look short and thick like this. But so in this first phase of cell division, metaphase, that's one of the things that happen. Those long DNA molecules, and these are molecules that are a couple inches long in every cell. As these, but they'll coil up, and so they look short and thick now. And because they're still duplicated, they're still the two identical copies of that chromosome are still stuck together from S phase, so it looks like the letter X, like two chromosomes stuck together. Those are called sister chromatids. Those two identical chromosomes stuck together. So that's what's meant by chromosomes condense and become visible. They coil up, become short and thick, so you can see them under a microscope. Spindle fibers emerge from centrosomes. Those two look like spiders. Those are called centrosomes, and that's part of the cytoskeleton. Remember, the cell has a cytoskeleton that's made up of protein fibers that run throughout the inside of the cell. These spindle fibers are just some protein fibers that will, you can see them starting to project out of those centrosomes, and they'll extend out and connect to each of those chromosomes. The nuclear envelope breaks down. So normally the cell has a nucleus. There's a nuclear membrane, nuclear envelope surrounding the chromosomes, and that disappears. It has to disappear because the chromosomes are going to separate into the two daughter cells. You can't do that if they're surrounded by a nuclear envelope. And so these centrosomes that are sending out those protein fibers, they're moving to opposite ends of the cell, like you see in the next phase. So then Next phase says chromosomes continue to condense. Kinetochores appear at the centromeres. That's just kinetochores and centromeres. That's just here where these two copies are stuck together. The two copies of each chromosome, and that's where those spindle fibers attach to them. Something long to. And the mitotic spindle microtubules are attached to the kinetochores. So now there are these protein fibers or spindle microtubules from the two centrosomes at opposite ends of the cell, and they're attached to every chromosome. Every one of those duplicated chromosomes. And those spindle fibers are what moves the chromosomes around in this cell that's getting ready to divide. So first it'll line them up right in the middle, halfway between the two the opposite ends of the cell. That's called the metaphase plate, sometimes called the equator of the cell. It's like the equator of the Earth, surrounding the middle. So you see all the chromosomes will line up in the center. Now that says the centromeres split in two. So the centromeres where the two copies of chromosomes were attached, they'll separate. Now we see the sister chromatids have separated. Now they're not. We don't call them sister chromatids anymore. Each one is now a chromosome. So they're being pulled toward the opposite ends of the dividing cell. So that's anaphase. In anaphase is when you see them start to separate. Might just see a little bit of daylight in between two lines of chromosomes. Now in the last phase, chromosomes have moved all the way to opposite ends. So now there's 23 chromosomes, 23 pairs of chromosomes on the left, 23 pairs on the right. A new nuclear envelope will form around each set of chromosomes. So a new nucleus. So now there's two nuclei, each one with 23 pairs of chromosomes. The mitotic spindle breaks down, so all those protein fibers that were moving the chromosomes around, they just break down and disappear. These steps here are all mitosis, dividing the nucleus. But what about the rest of the cell, the plasma membrane and the rest of the cytoplasm? That's called cytokinesis, when the rest of the cell divides. You see it pinching in half there. That's called a cleavage furrow. A furrow is just a groove that forms all the way around the dividing cell until it pinches all the way in half, and now there's two new cells, each one identical to the parent cell back in interphase. In plant cells, it's a little different. This is a human anatomy and physiology class, so we don't care about plants so much. But in plants, if you were a plant instead of a person, when your cells would divide, if you're a human, your cells divide, they make this cleavage furrow until it pinches all the way in half. And now there's two separate cells. But in plants, they'll form a plants have another layer of stuff around. In addition to the plasma membrane, they have a cell wall around that. And so when a plant cell divides, all the mitosis is the same, but then no cleavage furrow. It just makes a wall forms in between the two daughter cells. There's no pinching in half. Just a wall forms in between them to separate them. So there's diagrams at the top. These are micrographs here, or photographs through a microscope of the different stages of mitotic division. So if you see if you're looking through a cell through a microscope and you see the centrosomes that are moving apart, and you can maybe just barely see chromosomes. They're not because they're coiling up and becoming visible. But if you still see a nuclear envelope, then it's probably in prophase. If you see visible chromosomes and no nuclear envelope around them, it has disappeared in prophase. But they're not lined up in the middle yet, then it's probably prometaphase. If you see them, one stage that's really easy to see is metaphase, because the chromosomes will all be lined up right in the middle. You'll just see the very obvious dark line right in the middle. Just remember, metaphase, they'll all be in the middle. Then if you start to see a little bit of daylight in between two lines of chromosomes, that's anaphase. When they get farther apart, so you see two clusters of chromosomes at opposite ends of the cell, and the cell's getting long, maybe starting to pinch in half, that would be telophase. Telophase and cytokinesis usually happen together. So all this stuff, these first few phases happen before cytokinesis begins, pinching the cells in half. In lab, when we teach this course with an actual lab, one of the things we do is look at dividing cells under a microscope. So in the lab, we have one kind of a source of dividing cells is fish eggs. We have a lot of slides where the little slices of fish eggs were put on a slide, and they're permanently mounted on there. We buy them with that stuff already on it. But a fish egg has a little developing embryo in it, so there's a lot of cell division going on. And so you can look under a microscope and just see cells in various stages of cell division, and it's pretty easy to tell. This one's in interphase, this one's prophase, this one's metaphase. It's kind of fun to look at them and figure out what's going on in each one of those cells. Okay, so same thing, just more detail. And so these diagrams show there's only 2 pairs of chromosomes shown here. In a human, there will be 23 pairs, but it's simpler to see it with just two pairs of chromosomes. So here's the centromeres or centrosomes, and moving to opposite ends, and then they send these protein fibers or spindle fibers out that attach to every chromosome. The nuclear envelope disappears. So under a microscope, we might see, we're starting to see the chromosomes visible, but the nuclear envelope is gone. So that's probably in late prophase or pro-metaphase. So metaphase, very obvious. You see all the chromosomes are lined up right in the middle. And there's the two centrosomes and the spindle fibers attaching to them. It's pretty obvious if you see that under a microscope. It's no mistake. That cell's in metaphase and it's dividing. Anaphase, obvious anaphase, two lines with a little bit of daylight in between. So the two sister chromatids of each duplicated chromosome have separated and being pulled apart toward the opposite ends. This is backwards. We got early anaphase on the right and late anaphase on the left. So this is starting to look like telophase because they're pretty far separated. You see a cleavage furrow pinching in half. So there's telophase where the new nuclear envelope forms around each set of chromosomes and the obvious cleavage furrow. And this cleavage furrow forms, that's the cytoskeleton doing that. All these, they're not shown on here, but there's a lot of protein fibers, microfilaments, intermediate filaments, and of the cytoskeleton that pull the cell into that shape until it's completely separated. And cytokinesis, the separation of the rest of the cell, the plasma membrane and the cytoplasm. Now each of those daughter cells will go into interphase, starting their own life. Another summary of all of that in one big flow chart. And here's what these different stages would look like under a microscope again. Okay, so that was mitotic cell division and mitosis. Meiosis, or the reproductive type of cell division, is similar but different. It involves two divisions, one after the other. And we call those two divisions meiosis 1 and meiosis 2. Each one of those divisions, meiosis 1 and 2, have the same prophase, metaphase, anaphase, and telophase events, except they're a little different in some ways from mitosis, but the same kind of things happen. The chromosomes or the nuclear envelope disappears, the centrosomes move to opposite sides, send out spindle fibers to attach chromosomes, pull them apart into the daughter cells. But mitotic cell division is called duplication division because the daughter cells are identical to the parent cell and to each other. They have the same 23 pairs of chromosomes. But in this meiotic or reproductive cell division, the daughter cells end up being sperm and egg, and they have just one set of chromosomes, not two. They don't have 23 pairs of chromosomes, they just have 23 chromosomes. So each sperm and egg they're the only cells in your body that have only one set of chromosomes and not two. And then when they come together at fertilization, that fertilized egg will now have the normal 23 pairs, or two sets, one from each parent. So it's called reductional. this is reductional division because it results in half the number of chromosomes in the daughter cells. So the daughter cells, the sperm and egg, are genetically different from the parent cells because they have only half the chromosomes. And every sperm and egg that a person produces is genetically different. Every egg that you produce is different from every other egg, different genetically. Okay, so here's a summary of meiosis division. So here's meiosis 1 cell division, and here's meiosis 2. So just like before mitosis or mitotic cell division, in interphase, a cell that's getting ready to do this will duplicate all of its chromosomes. So there will be enough for water cells. So here's meiosis 1 division, but this is different from mitosis. In mitosis, remember the two sister chromatids, or the two copies of the chromosome separate into the daughter cells, but that's not what happens in meiosis 1. In meiosis 1, each pair of chromosomes separate into the daughter cells. So you have two number 1 chromosomes, two number 3, two chromosomes, two number 3 chromosomes, and so on. So they're shown as a red one and a blue one. So those would be so the two small ones at the top. You'd say, let's say those are the number 1 chromosomes. One of them goes into this daughter cell, the other goes into this daughter cell, but each one is still two sister chromatids stuck together. So the sister chromatids have not separated yet. But each of these daughter cells has just one set of 23 chromosomes now. So there's only one number 1 chromosome, one number 2 chromosome, one number 3 chromosome, but each one is still duplicated. Now in meiosis 2, this is where the sister chromatids finally separate. So this chromosome, in this chromosomes, the two sister chromatids will separate into the daughter cells. Now each of these 4 daughter cells now has just one set of chromosomes, 23 chromosomes, not 23 pairs like the original parent cell has. These will become sperm cells. If this happens in a male's testis, each of these will become a sperm cell. It'll grow a tail, a flagellum, become a sperm cell. So from one parent cell in a male's testis, meiosis will produce 4 sperm cells. They'll all be different. Each one will have different, will be different genetically. Same kind of thing happens in females, except in females, human females, there will only be 1 egg produced by this. Why not 4? Because in meiosis in female human ovaries, meiosis 1 happens just like in males, but one of these 2 daughter cells will die, just shrivel and die. It's called a polar body. Like it's in polar bear, called a polar body. So one of these will be a polar body and it will die. This other daughter cell will divide again and produce 2 daughter cells, and one of those will die. So this will be called the first polar body, and this will be called the second polar body. The polar bodies just shrivel up and die. So end up with only 1 daughter cell from meiosis. And probably the reason for that is that Well, in meiosis one, this polar body is very tiny compared to the other daughter cell. So this daughter cell gets almost all the cytoplasm and everything. This one has hardly anything in it. Same thing here. It's the one that survives is much larger. The polar body, second polar body, is real tiny. So this final egg that's produced by this is almost as big as the parent cell because these polar bodies are so tiny, and that's why a human egg is so big. It's a very large cell, so large you can almost see it with the naked eye. It's like a little tiny dot. But most cells are much smaller than that, and that's so because that's gonna that egg is going to get fertilized by a little tiny sperm cell that'll just deliver 23 chromosomes into it. But that egg has to be big enough that it can start dividing and form an embryo. Okay, something that happens in meiosis that does not happen in mitosis is called synapsis and crossing over. When in meiosis one, early in meiosis one, the chromosomes come side by side. The two chromosomes of every pair will come together side by side, like shown here. So the two number 1 chromosomes will come together side by side. The two number 2s will come together side by side, and so on. And that's called synapsis. It's word synapsis there. And as they are lined up side by side, they'll swap parts. Like the part of number 1 chromosome will break off, and the same part on the chromosome next to it will break off, and they'll trade. They'll just switch. They'll swap those parts. And so you can see they're different colors. The number 1 chromosome on the left is blue. The one on the right is red. But here, after crossing over, it's a mixture. And that can happen anywhere along the chromosomes. And sometimes it'll happen in several places along chromosomes. So because it can happen anywhere along those chromosomes, there can be an infinite number of different resulting combinations of genes in those resulting chromosomes. So that's one of the reasons that every sperm that a person produces is different from every other sperm, because of this crossing over that just happens at random places along the chromosomes. And it happens on all 23 pairs of chromosomes do this. Another reason for that every sperm and egg that a person produces is different is called independent of chromosomes. What that means is, for example, in this dividing cell, let's look at a different diagram. We'll show this better. Okay, when we look at this in meiosis one, we're just looking at 2 pairs of chromosomes in this diagram, but in this case we see that the red let's call the little ones number 1 chromosomes and the big ones number 2 chromosomes. So in this case, a red number 1 chromosome and a blue number 2 chromosome ended up in this daughter cell, and a blue number 1 and a red number 2 ended up in this chromosome. It didn't have to be that way. It could have been, this could have been a red number 1 and a red number 2, and a blue number 1 and a blue number 2, or this could have been a blue number 1 and a red number 2. This could have been a red number 1 and a blue number 2. So just with four, or 2 sets of chromosomes, there's 4 possibilities of combinations of chromosomes that could end up in a sperm or egg. But there's 23, because there's 23 pairs of chromosomes, and the number of different combinations that could end up in any one sperm or egg that a person produces is something like, there's trillions of possibilities. Just and that's why every sperm and egg is different. And that along along with the this crossing over, there's just an infinite number of different possibilities of, even though I have 23 pairs of chromosomes and each one has genes, when I make a sperm cell, only one of those chromosomes goes into each sperm cell, and so which one it is, which one of each pair is just random. So there's just the so there's that's why every sperm and egg is different that a person produces, and that's these chromosomes after this crossing over has happened, like we call them recombinant chromosomes, because the genes in the two chromosomes of each pair, the genes have been shuffled, recombined into the resulting chromosomes before they separate into the daughter cells that will become the sperm or egg. Okay. Any questions about cell division? Cell division is one of the most important concepts in biology. In every general biology class, spend a lot of time talking about the details of mitosis and meiosis. If you take a cell biology course, you'll spend weeks just on lots of details. You learn a lot more than we're covering in here. Okay, so, checkpoints. In all these steps of the cell cycle. For example, the DNA synthesis, copying all those chromosomes. Cells make mistakes doing that. The cell is trying to make an exact copy of every chromosome before the cell divides. But what if it makes a mistake? Each chromosome, remember, is a long DNA model. Remember, a DNA molecule is two strands of nucleotides, right? A, C, C, G, T, T, all these letters. I'm just making this up. And the other one is the mating strand, right? So, and these are hundreds of millions of these nucleotides in one chromosome. Just, these letters. So the cell is copying, trying to make an exact copy, every letter the same. You think cells ever make mistakes doing that? Yeah, they make mistakes. They might accidentally put a T there in the copy. That's what we call a genetic mutation, just a mistake. And some kinds of environmental agents can cause this to happen, like ultraviolet radiation from the sun comes in, hits your skin, penetrates into your skin cells, hits that DNA. It can cause changes in the DNA. It can cause a chromosome to break and then they might come back together. It might break here, and then it'll flip upside down and reattach the wrong way. Now that's a mutation, change. Mutation is just a change in DNA. If it happens in your skin cells, it can cause cancer. If it happens in mistake in meiosis, when you're making egg cells or sperm cells, then that could be passed on to your kids. If that egg gets fertilized with a mutation in it. So mistakes happen in cells. So if the cell makes a mistake copying, after the cell copies all of its chromosomes, it'll check. It'll compare the original to the copy and make sure that they match. It'll compare. It'll compare both copies, make sure they're the same. And if it finds a mistake, it'll fix it. It'll say, aha, that's not supposed to be a T, it's supposed to be a C, and it'll fix it. It'll replace that nucleotide with the correct one. But it doesn't always catch the mistakes. So these are called checkpoints. Just different stages of the cell cycle, the cell checks to make sure that it was right. What would happen if when these two chromosomes are supposed to separate into the two daughter cells? What if they both end up in this cell and this daughter cell, and this one doesn't get one of those chromosomes? It's a mistake. Right? That's another thing that the cell would check for that, would watch for that. Did that division happen correctly? Is there the right number of chromosomes in the daughter cells? So there's several of these checkpoints that happen at different stages along the cell cycle. If the cell finds a mistake, it'll try to fix it. If it's such a big mistake that the cell can't fix it, it might, this word apoptosis. This is like the cell committing suicide. So if a cell finds a mutation in the DNA and some cell in your body, mutations are what cause cancer. Cancer is just a cell that is dividing out of control. It doesn't know when to stop dividing, so it'll divide, and then those daughter cells will divide, and they'll divide, and none of those cells will become normal cells. All they're doing is dividing, making tumors, and disrupting the normal cells in your body because of a genetic mutation in one of the genes that has the information for when the cell should divide or not divide. So if the information that tells the cell don't divide right now, that gene doesn't work anymore, the cell doesn't know when to stop dividing. So and some cells will if there's a mutation, they can't fix it, the cell will just commit suicide rather than keep dividing and passing on that mutation into more daughter cells that are not normal. Of course, this doesn't always happen because people get cancer and genetic mutations get passed on to kids all the time. So uh, this word apoptosis, it generally means just a cell dying. Some cells are just genetically programmed to die after a certain period of time, but apoptosis can be because of some reason that the cell decides I'm going to commit suicide for the good of the entire organism, the entire body. Okay, some practice questions and some videos, links to some videos that might be worth your time to look at. These are pretty short, most of them. So it's, I'm trying to explain stuff with just pictures, but if you watch a video of showing this stuff happen, it's much easier to understand. But I think we need to move on to the next chapter. Four. Chapter 4. Okay, get started on this chapter. So finally, we're actually getting into some anatomy. So we're done with that chemistry and cell stuff. Now we're actually getting into some anatomy. So this chapter is about tissues. Each chapter will be about an organ system. Chapter 5 is about the skin, integumentary system. But all organs are made up of tissues, so we need to learn a little bit about what tissues are, what are the different types of tissues that make up organs in our bodies. Definition of a tissue is just a material that makes up our bodies. And this material is made up of cells, and most tissues have non-living stuff in them as well as the living cells. And that's called the extracellular matrix. It's the non-living part of tissue. Extra means above or outside of. So extracellular means outside of the cells. And that extracellular matrix is, well, we'll see what that's made up of. But there's 4 basic types of tissues or categories of tissues listed there: epithelial, connective, muscular, and nervous. So we'll look at each of those a little bit. These two we're just going to look at pretty briefly because we're doing a couple whole chapters on this, and I think 3 or 4 chapters on the nervous system. So we'll get into that in more detail. But these first 2, epithelial and connective, we'll look at them in more detail. Okay, there's some diagrams of typical epithelial and connective and muscular and nervous tissue. Epithelial tissue is tissue that's in thin sheets, thin layers, like this what your skin is. And your skin is epithelial. The inside of your mouth, the lining of the inside of your mouth is epithelial. The coverings of your organs, the lining of hollow organs, that's all epithelial tissue. In this diagram, we see these are tall, skinny cells side by side. And then below that, that thin blue layer That's, in the case of epithelial tissue, that's usually where the extracellular matrix is. So there's a layer, one or more layers of living cells, and then right below that, that's non-living extracellular matrix. Connective tissue, that looks like a confusing mess of stuff going on there, but the cells, that's basically cells and a few different kinds of cells, and those long things like threads, those would be protein fibers that add strength to the connective tissue. A lot of different kinds of connective tissues that we'll talk about. But muscular tissue, muscle tissue can do something. It's specialized to do one thing. Muscle tissue can get shorter and produce force. This would be the muscle tissue of skeletal muscle, like the kind that moves through joints. So the muscle cells are long, like spaghetti, like this. Nervous tissue, that would be a neuron or a nerve cell. Looks like something from a science fiction movie, a one-eyed monster or something. Okay, some examples. It's a kind of a blurry picture. Nervous tissue, the brain, spinal cord, and all the nerves throughout your body that connect or communicate different parts of the body with each other. Muscle tissue, we'll see there's three types of muscle tissue. Epithelial is everywhere in our body. We have epithelial tissue because most of our organs have some kind of covering or lining that's epithelial. Connective tissue, it's everywhere. Lots of different kinds. Your bones, cartilage, blood, even blood is considered to be a connective tissue. Fat tissue is connective tissue, as we'll see. Okay, again, brief definition of each type. So epithelial, sheets of cells that cover surfaces. We'll mention that the glands, most glands have a type of epithelial tissue that produces whatever hormone, or whether it's producing sweat or mucus. Connective tissue binds cells and organs of the body together, protection, support, integration of all parts of the body, and so on. Okay. In a developing embryo, each of us starts out as a fertilized egg, and then that divides a bunch of times. It becomes a solid ball of cells, and that becomes a hollow ball of cells, and eventually it starts to look like a little person. But very early on in a developing embryo, there's 3 layers of cells. There's only 3 layers of cells, and they're called the ectoderm on the outside. The endoderm is the deep center part, and then in between is the mesoderm. And so every tissue in your body started out as one of those 3 layers of cells. And so we know that epithelial tissues originate in all 3 layers. For example, your skin started out as ectoderm because ectoderm is on the outside, and that's where your skin is. But there's also other epithelial tissues originate in the endoderm and mesoderm. But nervous tissue originates mostly from the ectoderm, and we'll see how that happens when we get into the nervous system later, toward the end of this semester. And muscle tissue and all the connective tissues come from the mesoderm, so that's the in-between layer. Um, okay, membranes. Before we get into this, the epithelial and connective tissue, we need to um, talk a little bit about membranes. Membranes are not one or the other. They mean both epithelial and connective tissue. It's flat sheets, pliable tissue that cover or line a part of the body. So it's an epithelial tissue because it's in thin sheets, but it also usually has connective tissue to give it some strength. Types of membranes, epithelial that are mostly epithelial are include mucous membranes, serous membranes, and cutaneous. So the word cutaneous means skin. So cutaneous membrane is skin. Mucous membranes, we have lining most of our digestive system, starting in your mouth. Your mouth is lined with mucous membranes, and your throat, up your esophagus that you swallow through, your stomach, small intestines, large intestines. That's all lined with mucous membrane. Mucous membrane is just thin sheets um, uh, of tissue of epithelial cells, and in among or between the epithelial cells there are mucus cells that produce the mucus. Um, and serous membranes, that's what the pleura, pericardium, and peritoneum that we learned about in the first chapter. Those are called serous membranes. Um, Membranes that are mostly connective tissue, they're called connective tissue membranes, and um, these are, um, some important ones are the ones that form joint capsules around your major joints of your body. They're called synovial capsules, and um, um, they so your major joints like your elbows, knees, shoulders, uh, hips, even finger joints. Each one has where the bones meet to form those movable joints. There's a capsule surrounding that joint made of this kind of connective tissue membrane, and that membrane secretes a slimy liquid for lubrication called synovial fluid. Um, for the joints to move smoothly. Okay, just more about the examples of membranes in the body. Um, Cell junctions are, the word junction just means where two things come together. So cell junctions are connections between cells in epithelial tissue. Just, epithelial tissues are typically cells in layers, and those cells are side by side. So what holds those cells together in those tissues is these cell junctions. And there's three main types: tight junctions, anchoring junctions, and gap junctions. And they're shown here in these diagrams, but described here. Tight junctions, the main thing about tight junctions is that they are watertight seals. They form a watertight seal and these cells side by side to make up the epithelial that lines certain organs that contain liquid, like the stomach and the intestines and the urinary bladder. Those cells have to be held tightly together, side by side, so no liquid can leak in between them. You don't want stomach contents leaking out into your abdominal cavity. That's strong acid. You don't want urine leaking out of your urinary bladder into your abdominal cavity. Anchoring junctions provide structural connection between cells. This holds them together. And they're called anchoring junctions because they are protein molecules that are like anchors between cells. So here's two cells side by side in epithelial tissue. These protein molecules that extend through the plasma membrane of both cells, and they form anchors, holding them together. And there'll be a whole bunch of these. So the desmosomes and adherens junctions are like this. And the difference between those two types of anchoring junctions is just what kind of protein molecules are doing the anchoring. But this other one, hemidesmosomes, remember I said epithelial tissues are usually made up of cells side by side, and then right underneath that is this, it's called a basement membrane, the extracellular matrix. Well, the hemidesmosomes are, instead of anchoring cells together, they're for anchoring these cells to the basement membrane. So same kind of protein molecules that do this anchoring. Those are the hemidesmosomes. And gap junctions are not so much for holding cells together, but they make little tunnels from one cell to another. So if there was a gap junction here, it would form a little tunnel, so that stuff could move from one cell to the other. So those are called gap junctions. Okay. One main difference between epithelial tissue and connective tissue is epithelial tissue is mostly cells, and the extracellular non-living part, if there is one, it would be in a layer below the layer of cells. Like this is, I think, skin, cross section of skin. So it's all cells. It's all skin cells, and then below that there would be a basement membrane of non-living stuff. This is connective tissue. It's mostly non-living stuff with just cells scattered throughout here and there. So mostly extracellular matrix, and this is mostly cells. What else is it? Less vascular. Epithelial tissue is less vascular. Vascular means blood vessels. So epithelial tissue doesn't have as many blood vessels running through it, whereas most connective tissue does. So it's more vascular. Okay, some sample or practice questions here. Okay. So more details about epithelial tissues. Widespread, so they're everywhere in the body, sheets of cells. Protection, secretion because, well, for example, your skin is an epithelium, and it secretes sweat and oil, and the epithelium in your ears is where the earwax is secreted. So, and some epithelia produce mucus, so that's secretion. Permeability just means that stuff can pass through it. So we can absorb some things through our skin, but we have to be careful about what we contact. There's some harmful toxins that they get on your skin, they'll penetrate right into your tissues. Excretion means getting rid of waste products, and sweating, perspiring is one way of excretion, get rid of excess water, but also a lot of salts and urea, some waste products in sweat. Sensory reception, we especially in your skin, you have a lot of receptors for hot and cold and pain and pressure and touch. and vibration. That's what's meant by sensory reception. Uh, epithelial typically have a nerve supply, but don't have uh, blood vessels within the epithelial tissue. And mitosis occurs frequently to replace cells that slough off, or so your skin, for example, constantly making more. The lining of your digestive tract, you've got food moving through it all the time and rubbing against it. So, um, especially in your stomach, it's very acidic in there, hydrochloric acid in there. That's really hard on those cells lining your stomach, so your stomach is constantly replacing them, making new ones. Um, okay, these many different kinds of epithelia that we have in different parts of our body, um, they, the, they can have different shaped cells. Cells can be thin and flat, or they can be box shaped, or they can be tall and skinny. And so that's what's meant by these epithelial cell shapes. It can be, this word squamous means flat cells. Cuboidal means box shaped or cube shaped cells. Columnar cells are tall and skinny, or tall and rectangular, shaped like a column. And these transitional, the word transition means change. So these are cells that can change shape. And these transitional cells are in tissues that stretch, like um, in the urinary bladder. When it's empty, it's small, but when it fills with urine, the tissues surrounding it has to stretch. So the cells change shape as it stretches. So that's would be transitional. So let's look briefly at these cell shapes. So squamous just means thin and flat. So we're looking at cross sections through a layer of thin flat cells. These would be the cuboidal, obviously shaped like a box. These tall thin ones, columnar. Um, so we use those words to describe epithelia. Does it have squamous cells, cuboidal, or columnar? Um, and also we use the words simple or stratified. Simple, if we're talking about epithelial tissue, simple means just one layer of cells like that. So we call that simple squamous epithelium. If there's two or more layers, we call it stratified. The word stratified means in layers. A stratum is a layer of something. So this would be stratified cuboidal, stratified columnar, and there's one other. This one's kind of weird. It's called pseudo-stratified. Pseudo means false. So somebody decided that we should call this pseudo-stratified because it kind of looks like it's stratified because this is the nucleus of each of these cells. And then the nuclei are at different levels. So it kind of looks like it's stratified because the nuclei are at different levels, but it's just one layer. So we call it pseudo-stratified. Looks like it, but not really. And some of these, you have this looks like a brush, bristles of a brush. These are the cilia on this cell. Remember those finger-like projections sticking up, and so some of these pseudo-stratified and columnar cells have this. These are cells lining or airways, have these cilia um, at the tips. Some terms here that are used referring to cells of epithelial. So the lateral surface would be the sides, where one cell meets another. The basal surface is at the base, the bottom. and then the apical surface is the top, the tips. The tip of something is called the apex, so it's called the apical, so it would be the apical surface of these cells. Okay, so combining the shape of the cells and whether it's one layer or more than one layer, we come up with these different combinations: simple squamous, simple cuboidal, stratified columnar, transition. So again, these are the ones that change shape, so they stretch. This is epithelia that stretch and then shrink back. Glandular glands, most glands are made up of epithelial tissue. It's usually columnar cells that form a hollow tube, and then those cells produce something, sweat or hormones or something, into the tube, and then that tube leads to either outside the body or into the blood uh, to put hormones into the blood, or whatever. Okay, so these two slides here just list the different types of epithelial tissue and some examples of where they're located in the body and their functions. So like this simple cuboidal, like I was saying about glands, they secrete stuff in glands, secrete hormones or sweat or earwax or whatever. Simple squamous, that's just one layer of thin flat cells. So these are in places where maybe diffusion or epithelial epithelium is important. For example, in essentially the air sacs of the lungs. It's the first example there. We inhale air with oxygen in it. That oxygen has to diffuse from the air sacs in your lungs into the blood, in the blood vessels in your lungs. And so the epithelial tissue that oxygen has to diffuse through, it's better if it's thin and flat, so it doesn't have very far to diffuse. So those air sacs are made of simple squamous epithelium. Yeah, blood vessels, same thing. We have to have oxygen and nutrients diffusing or moving out of your blood into your tissues all the time. So the capillaries of your blood, which are the tiny blood vessels where that movement in and out happens, simple squamous epithelium. I won't go through all of these. Stratified squamous, many layers of thin flat cells. That's what your skin is, and so it's many layers. It's a bottom layer is where those cells are always dividing to make new ones, and then they move up through the layers. And the top layer is the layer on the surface that's always rubbing off, so they have to be replaced all the time. It's a transitional that as this tissue stretches, these cells become longer and flatter. So we have that lining the urinary bladder, the urethra and ureters. Those are the tubes. The ureters are the tubes that go from either kidney to take the urine into the urinary bladder, and the urethra is the tube that goes from the urinary bladder to the outside, where when we urinate. Glandular epithelium. A gland is an organ that secretes something, either hormones or earwax or sweat or saliva or digestive chemicals for the stomach or intestines. Mammary glands make breast milk. And there's two kinds of glands, and we'll spend a whole chapter next semester on endocrine glands. That's the hormone system, the hormones, endocrine glands. They make hormones. Those hormones go into the body fluid, usually go into your blood. But the exocrine glands make something that it does not go into your blood. It makes something that either is released onto the outside surface of your body, like sweat or skin oil, or it will lead to some hollow space inside your body, like saliva. Salivary glands release saliva into your oral cavity, your mouth. We have, the pancreas makes digestive chemicals that go into the hollow part of the inside of the small intestines. Mammary glands release breast milk to the outside. And but these glands are usually in the form of this would be a cross section through a tube in a gland, and so these would be cuboidal cells, epithelial cells surrounding that tube, and these are the cells that would make the stuff, whatever it is, breast milk, sweat, hormones, that would go into that tube, and that tube would take that stuff to, if it's sweat, it would take it to the outside of your body. If it's a hormone, it would lead into your, release it into the blood. So this example is a sweat gland. It's just tubes surrounded by cuboidal epithelium that produce the sweat into the tube, and then this tube would lead to the surface of your body where the sweat is released onto the surface of your skin. There are some glands that are just one cell. That's what the so-called goblet cells. So this is called a goblet cell. So there would just be these in among the cuboidal cells of mucous membrane epithelium, and they produce mucus. But this is what glands look like, and they're different shapes. Some are just a simple dead end tube like these, and some of them have branches. But these are all just tubes surrounded by cuboidal epithelium that's producing something into the tubes, and then the tube takes it somewhere. Let's stop here. That's enough. We got about halfway through. So I'll go try to grade these things right now, and if I can, I'll get the grades on Moodle right away. You guys feel okay about it? It wasn't too bad.
Grade back for the test.
I'm going to try to get them on Moodle this afternoon.
Okay.
Yeah, so check in an hour or two.
Okay.